So, let us start the 40th lecture today, which is on silk biomaterial and electricity. So, in the previous lecture, I shared the story of Jacobus Ishay and his exploration of understanding the thermophotovoltaic properties of hornet silk cocoon membrane, and this, which is also called the silk cap. So, today we will be talking about the other cocoon membranes, which is the more traditional ones, the Bombyx mori and the Tassar cocoon likewise. And we will see whether the observations which were made by Jacobus Ishay during 1970s, 80s and 90s with his seminal work, do they really hold true for more commonly known silk, or is it just a very unique property of the hornet nest? So, as I told you in the previous lecture that in one of the studies it was observed that the regular silk cocoon has a thermoregulatory property. That gives a positive hunch that possibly if it has a thermoregulatory property, it may have certain currents across its membrane. So, this lecture will be to explore that and further whether these kind of hairy currents or very small current, could these current be utilized in the era when we are kind of facing a lot of energy crisis as we are looking for different kind of materials to generate power. Could these kind of naturally existing material could be utilized to power some low power electronic devices or not. So, this study will take us from Ishay's original study on hornet nest to the next level of study, where we will be dealing with the silk cocoon membranes of Bombyx and Anthrea mylitta. Okay. So, this is a repetition of the picture what I showed you people. So, if you look at these two cocoon, so on your left hand side with the A is a Bombyx cocoon. Bombyx is the most commonly used cocoon for the garments across the world. China being the largest producer, followed by India, and this cocoon is very smooth cocoon and very smooth. This silk is very smooth, and it does not have a lot of minerals. It has very little mineral constant contents, and that is why it is so smooth and so fine. Whereas the second one on the B, what you see Anthrea mylitta or the Tassar cocoon, this is also called a wild cocoon because it grows in the wild or sometimes semi domesticated kind of So, this cocoon has lot of mineral contents, and that is a challenge in order for the reeling of the fiber, because of the lot of mineral contents. This cocoon is kind of a coarse, it has a very coarse texture as compared to the texture of the Bombyx mori cocoon. So, this lecture will be talking about the electrical properties of these two types of cocoons. So, let us look at the ultrastructure of these cocoons. So, this is the paper I will be referring to. So, I will be adding this paper in your additional material, reading material. Please go through this paper. So, all the studies what is being shown will be from this paper, and so that sums up. There are 3 papers what I expect you from the previous lecture, those 2 papers from Jacob Ishay and this paper from Scientific Reports. Please go through them. Okay. Now, let us move on to the ultrastructure. So, if you look at the ultrastructure of the silk cocoon membrane, on what you see in A is the ultrastructure of Anthrea mylitta or the Tassar silk, and the B what you see is the ultrastructure of the Bombyx mori cocoon. So, if you look at the structure very carefully, you will observe in A and B, the basic difference is A has lot of as if the granules scattered on those sericin fibroin network, whereas you see a very smooth texture in the case of Bombyx. Now, the upper two panels are showing the outer surface of the silk cocoon. If you see the inner, because it has been cut into two halves. So, that what you see here is the outer surface, and what you see here is the inner surface, both inner and the outer. So, the lower two, C and D, are the pictures of the inner surface, which is fairly smooth in both the cases. But in terms of the fiber thickness, the fiber is slightly more thicker in the case of the Anthrea myelata, which is wild type cocoon, which is also called a tassar cocoon. And A and B, which is the outer surface of the cocoon, what is being shown. You see the threads are coarse as well as in the case of Anthrea, and you see there is an inset box which is showing some, in the left hand top corner, if you see it, those are showing the crystals. What you see, those small, the granules all over the surface, it is showing the crystals of calcium oxalate. They have enormous deposition of calcium oxalate on their outer surface. Okay? So, this is the basic ultrastructure, and the basic ultrastructure still follows the same what I told you, that inner cylinder, inner core cylinder of fibroin. This is the fibroin core, and this is the gummy part, which is the sericin or the sticky protein. And on top of the Anthrea myelata, what you see a lot of sticky crystals which are present here. And these sticky crystals are of calcium oxalate, what you see in the inset picture. So, basic geometry is the same. There is no difference in the basic geometry, except there is one more change. What will you observe in the next slide is this one, the amount of elements which are present in it. This was also observed by Jacob Ishay while talking about the silk of the hornet nest. So, if you look at it, there is significant proportion of calcium, sodium, potassium, chloride, magnesium, sulfur, and copper. Whereas in the Anthrea also you pretty much see the same thing, except they have zinc, which is in significantly higher amount, at least not detectable in Bombyx, and they have phosphorus, which is not detectable in Bombyx, and of course, the sulfur and all those things are present, and the copper, which is not detected in the case of Anthrea. Okay. So, if you look at it, so there are plethora of, and the calcium is higher in one of them, which is in the Anthrea. Calcium is fairly higher, and that calcium possibly is coming from the calcium oxalate crystals, because there in the Anthrea there is enormous calcium oxalate crystals, and that is why the concentration of calcium is very high in case of Anthrea myelata. Okay. Now, if you look at this, so it means what we are talking about, now I am adding one more component to it. So, they have lot of these X are showing the different kind of elements which are present there. And one more thing here I want to add. All the studies made by Ishay was when you are keeping the cocoon at a slightly higher relative humidity, at around 80% relative humidity, 80% to 90% relative humidity, you are observing all the different currents which are observed by Ishay. It means the ambient water plays certain critical role in charge transfer. Okay. So, now moving on to the next slide, in order to understand how the cocoon properties could be studied, you have to develop certain very specific standard structures. So, what has been done in this study is, you took two different cocoon samples, you took a piece of Bombyx, and the measurements are given out here. They have a slightly varying thickness that you can't help it because it is inherent property, and you connect it with two different electrodes. So, here let me add, they have a certain amount of voltage across the cocoon, across the membrane, but that voltage is not very high as was observed by Ishii. So, in order to enhance that, so silk cocoon has its inherent potential difference, okay, PD1, which is indigenous potential difference. And now, by putting two different electrodes, say an aluminum and copper, you are adding one more potential difference to that, which is the second one. So, the total potential difference is now slightly higher. So, now what you are using it in order to understand, because now we are more keen here to see whether this could be used to develop some kind of device or not. We know that there is a flow of current and all those things, but could this be utilized? Okay. So, in the next study, what has been done, by developing these kind of devices, the current and voltage are being measured. Now, let us see how the current and voltages are changing. Okay. So, if you look at a dry cocoon, now follow, this is a busy slide, go to the panel A. If you see in the panel A, in the case of a dry cocoon, there is very little current. You could see that the current is very less. Okay. Now, if there is a moist cocoon, then the current jumps very significantly. Follow the only the panel A. Now, if you demineralize the cocoon, then also you will see a significant amount of current. Demineralization means you are getting rid of all these X, you are getting rid of the X out here. So, it means you are getting rid of several charge carriers out there. Now, the current density increases significantly if you see the picture when you are exposing this to water vapor. Okay. And if you dope it, now you are adding NaCl to that, a kind of ensuring when the coming in contact with water, what it will do? It will form Na plus ions and Cl minus ions. What you will observe is the current density goes up. So, same thing in the case of Anthria. If you follow the section C, you see the same thing. It pretty much follows the same trend. Dry cocoon, it is almost acting as an insulating material. There is hardly anything. But the very moment there is exposure to moist condition, this behaves as if it is a conductor or something like a semiconductor, or almost behave like a conductor. Okay. So, if you remember, I was telling you all the Ishii's work follows that relative humidity is at 80%, or at only at relative humidity 80% you see this kind of charge motion. But then you have to realize most of these hornet nest or silk cocoon remain in an area in kind of in a micro environment where there the humidity is higher. They may be in a shade in a corner, or surrounded, camouflaged by several trees or something, the leaves likewise. So, those kind of camouflaged regions have a micro environment where the relative humidity is slightly higher, because there is a transpiration taking place continuously from the leaves and the surrounding vegetation, and that kind of increases the relative humidity in that location. Okay. So, now if you see the voltage trend, you again see the same thing. At a dry, the voltage is fairly low, but then as you moisten it, the voltage goes up. It means one thing is very clear, that this kind of situation are wet electricity. This is not something so. Still the question remains, who are the charge carrier in this kind of situation? Okay. So, this is the first clue in this study, that this kind of materials only functions when it is exposed to, or it is kept in a kind of high relative humidity. Now, this is a very interesting study to look at. What are the thermoelectrical properties of it? So, if you follow this graph, if you follow the, let us follow, because the trend is same, let us follow in the case of Bombex the red line. If you see it, as you are moving, the red line is showing you the current, as the temperature is increasing at around 45. So, there are two rise you are seeing, one you are seeing between 10 and 25, and then second you are seeing between 45 and 60. So, around 40, 45 or so, beyond 40, slightly at around 50, you see a sharp spike of current. Okay? And you saw such sharp spike of current in case of Ishi's work at around 31, 32 degrees centigrade. Here you see it at around 45, 50 degrees centigrade. Okay? There is a sharp rise spike of current. So, there are two spikes of current and you see the same trend. So, it means this kind of membranes are thermosensitive membranes. First conclusion you could draw, and during the this thermosensitivity leads to generation of electrical currents. This is what this slide is telling you, and this trend is pretty much the same in the case of Bombex as well as Anthria. So, what I was trying to explain in the previous lecture, in the 39th lecture, that these membranes have a thermoregulatory role which is governed by flow of thermal current, holds true here. If you see that depending on the temperature shifts, the thermal current is varying as well as the voltage. So, it has certain thermovoltaic property which leads to thermal current generation. Okay. Now, if you see the IV characteristics, and once again for those who have joined late, like you can see all these figures in the reference paper what I have given you. Okay. Now, if you look at the IV characteristics, you will see the current voltage pattern shifts or changes with respect to temperature. So, there are two IV thing which has been taken at 30 degrees centigrade and 80 degrees centigrade. And if you see it, the current density is changing as the temperature is rising. And this exactly will remind you what Ishe did. If you remember, I will just go back to the slide of Ishe is here in the hornet silk. It is following the very similar trend what you have seen in hornet. The current rises when the temperature rises. Okay. So, coming back to the IV studies, that is what is the IV study is telling you, as the temperature rises, the current rises. Okay. Now, followed by the next study. So, one of the thing is, one of the possible explanation to this current could be, now this is all we are trying to figure out, could be all these different elements which are present there in the presence of water vapor. So, the water vapor is getting embedded into these zones. Okay. I will show you a wonderful model afterward. Okay. So, these water vapors are, so they are creating different kind of electrolyte materials. There could be sodium, there could be calcium, there could be magnesium, likewise different charges, there will be chloride, there are other anions and cations out there. And now, since these anions and cations are sitting across two electrode of different electronegativity, there is a possibility that what we may see a charge flow. This could be one explanation, but this explanation fails if we look at this picture. Why in the case of demineralized condition? If you see this picture very carefully, on the second panel A, see the panel A and the orange line, a demineralized cocoon and a moist cocoon. You see the same situation. Why is it so? Because when you are demineralizing, you are essentially getting rid of all these things, getting rid of all of that, minus these. So, then where are the ions? So, that is a very intriguing question, and that is what we are going to go slowly, that what possibly could be the explanation. Because again, coming back to the slide, be careful with those, that orange line and the violet line just underneath it. You see that orange line which is showing demineralized and getting WV as the water vapor and the moist cocoon and just a water vapor. So, those three lines are very important, the green line, the orange line and the violet line. These three lines are telling you something, and we will be talking about what these three lines are telling you, because in those three lines lies some of the very fundamental phenomena which is getting orchestrated in between this membrane. Okay. So, coming back, in order to see, so this is an NMR study which was done to see the presence of the water molecules which are present embedded in the cell fibroin matrix, as well as the presence of the sodium ions which are present there, and those could be detected using the NMR out here. So, it is kind of to show that there are ionic species which are present there. Now, followed by that, we have to went ahead and see whether these kind of things. So, we have observed that there are, you see these kind of currents are present there. Okay. So, at this stage there are two things what we will be dealing with. What we will be dealing with is that whether these currents are sufficient enough, what you see in the slide out here is sufficient enough to drive some crude devices, or could you develop some bioelectrical device to harvest necessary energy from a cocoon or not. This is one thing what we are going to answer. Second thing we are going to answer, what are the charge carriers. So, I will be answering this charge carrier thing in the end, but first I will be discussing about what are the different devices you can develop out of it. So, here there is some device development we are talking about. Okay. So, you take the cocoon, you could see in the picture very clearly, the inside you see there is an aluminum foil which is acting as an electrode, and outside you see the wrapping of the copper. So, you have the aluminum and copper electrode which are attached across the cocoon surface. Okay. So, now what you see here, now when you have this aluminum copper and you are, so let us concentrate on panel A, B, C, D. Okay. So, you have three cocoons out here which are attached by aluminum and copper, and they are attached in a series circuit, and you are exposing this to water vapor. You will see it could generate sufficient power to power an LED, light emitting diode. Whereas, you can do the same thing, but we have just shown it here with 4 different Anthrea mylitta cocoon. If you connect them in series, exactly the way it is, you can really glow an LED out of it. So, you can harvest energy by exposing a cell cocoon to water vapor, and water vapor which is at a temperature around 80 to 90 degrees centigrade. Okay. So, that is the ambient temperature. So, underneath what you see that there is a beaker coming in the panel A. From that beaker, basically beaker is getting, it's a boiling water which is the water is boiling and the water vapor is coming out, and in the presence of water vapor what you see is that the cocoons have started to glow. Okay. So, now the question arises that could we really make a very this is a very crude device which is exposed to water vapor and likewise, but could we make a much more finer device. But before we get into this, let us look at these two panels, E and F, what the E and F is telling you. Okay. One more thing I needed to add, you can read the paper, the reference paper, it is free of cost online, and you will come across a series of videos where you will be able to appreciate the whole process, how these devices are being used to generate electricity. You can actually harvest electricity from these devices, and please go through those videos, because that you will be able to appreciate the whole process much better as compared to seeing a still picture out here. Now, what about this E and F? What E and F is telling you? E and F is there is no water vapor or anything. It is a moist cocoon which is charged for a small period of time using a charger. You have these battery chargers or something, you charge it and then disconnect it. Then you connect a load on it. You see an LED which is glowing. So, now you can switch it on, switch it off. So, it means the cocoon membrane what we are talking about, these proteins which are the assembly which is making the cocoon membrane is acting like a capacitor. or a simple charge storage device. Okay. This one you have to really see the video, then you will be able to appreciate that you can switch it off, switch it off, switch it on, switch it off, and you will be able to store sufficient amount of charge in them, which could be utilized for plethora of applications. Okay. So, this is a functional device from the crude cocoon, directly using the cocoon by the connecting the electrode like this. And what you see out here, you see these two tubes which are coming through. So, you can observe this by exposing it to water vapor, both outside as well as inside. What does that mean? So, let me just rub the board and tell you exactly how this is being done. I will just keep that relative humidity out there. So, if you have these cocoons connected in series, and what you do, you have a tube coming like this. That is what you observe, Cocoon 1, C1, C2, C3 likewise, and you are giving water vapor like this. So, there are two ways you can do it. Either you expose the water vapor like this from outside. What you are seeing in that image is something like this. If you see the slide, so in the panel A, what you are observing is the water vapor is being exposed outside, and you can even expose water vapor inside like this. And when you are exposing it to the inside, then also you see the same effect, but this effect is more pronounced because your water vapor is concentrated inside. So, it is kind of confined at a space. So, you see this effect much more elegantly out there, that it generates sufficient power. Okay. Now, coming to a systematic device development. So, here you see that the 4 cocoons were added by connecting with these connectors and likewise, but here what is being done, it was systematically stacked using 2 different kind of electrodes. Okay. So, this is what you see now, where there is a systematic device, and a systematic device also, if you see the videos, you will see, you can see the same kind of current and voltage. You will see these are the current traces, what you see, as in the presence of water vapor over a period of 200 minutes, which is almost 3 hours, you see significant amount of current, and you see the subsequent voltage in the bombyx as well as in anthidium. So, what I essentially wanted to put across is that this membrane behaves in a very, very interesting way, very similar to what has been documented by Jacobus Ishay, and that also gives a lot of confidence to tell that what he observed some 40, 30 or now almost 30 years back is pretty much the same what we are observing today with much more So, it means the silk proteins across the genera probably behave like this. They have, now we can say with certain degree of confidence that at least 3 of the silk species are showing the same behavior. Okay. So, these are the current and voltage of the traces of the devices. Now, we talk about the model. What is the proposed model, currently accepted model? So, you see here there is a temperature gradient shown at the top, okay, from 10 to 60 degrees centigrade and likewise and above. Now, cocoon membrane has been shown as the outer membrane and the inner membrane. You see this. So, you see in the center the outer membrane, and if you see the outer membrane has been shown as broken lines. It is shown in broken lines because the water absorption from the outside is higher, and the inner part is very, very insulated to water. It is almost like a waterproofing membrane. So, if you really could, this is the cocoon membrane. So, for example, this is the membrane cross section you are observing. So, there will be a lot of bigger pores here through which the water will enter. and but this pores density changes like this. By the time inside is very, this is almost like a waterproofing membrane. This outside, so this is the kind of gradient you see. These are showing the water molecules. So in other words, this membrane is an asymmetric membrane, and because of its asymmetry, you connect an electrode like this, you see a very interesting potential drop across it. And in this asymmetric membrane, there are embedded, metal ions which are present there, anions and cations, which are present there. And in the presence of water, these ions gets activated. So if you see in this picture, in the slide, you will see there is this ions and the water molecules in blue, and then you have the ions which are all scattered across it. And when it is getting sufficient water, those ionic species, based on the electronegativity, started to flow across the membrane. But what is interesting to note, now I will take you back to this slide, what I was telling you, that what is the explanation to this situation when you are demineralizing the cocoon. Still, you are seeing the same current. I told you that please concentrate on the green, orange and the violet line. Why they are same? They are pretty much same. This is something, a very mysterious situation. So it means there is something else probably. So even if you remove this, remove these red ones, still you see sufficient amount of current. How you explain this situation? This situation could be explained in a totally different perspective. That is something called proton hopping. What essentially happens is, even if you get rid of all these different ions, this particular temperature zone, there is huge amount of, at around 80 degrees centigrade, there is a huge amount of movement of the water molecules which are taking place on this surface at a temperature of 80 to 100 degrees centigrade. Enormous movement of water molecules. And these water molecules are moving, and this is already hydrated. There is a lot of proton species which are formed, protonic species which are formed there. And those protonic species possibly could, using the word possibly, could explain this unusual pattern, that even if you have a demineralized cocoon with you or a membrane with you, there is no mineral, still you see a high density of current present there. So it means this whole wet electricity what we were discussing is governed by two components. One of them is ion, which is a charge carrier, anions, and one second, let's go back to the slide, anions and cations, and the second one is protonic. Possibly these are the two things which are governing the flow of current across this membrane. This is the most possible explanation, but that takes us, it means whether the other kind of membrane could do the similar things. And as a matter of fact, if you go through the paper, what I have recommended you people, you will see some of the porous membranes like paper, if they are sufficiently doped with ionic species, they could also conduct current. This was a backup or kind of supporting information what you will see in the paper. Okay. So, coming back to the slide, if you look at the slide, so if you see a dry paper is acting as an insulator, a moist paper also does not show a lot of current, but in the case of water vapor you see a rise in current, and if you dope it with NaCl you see even a higher current across it. So, these studies and this model, and if you go through this slide where I was talking to you about the green line, orange line and the violet line of current, tells you that this is a very interesting zone where both ions and possibly the protonic species are responsible for the flow of current across these kind of biological membranes. So, partly where we started in the previous lecture while talking about Ishi's studies, what are the charge carriers? We partly answered this question in this lecture. Possibly the charge carriers are either ions or the protons, protonic species. Okay. Could we call it a biological semiconductor? Well, I won't be able to answer that question. It's a material when it is dry. These kind of biomaterials, when they are dry, they are insulator. But in the presence of water and varying temperature, their behavior drastically changes, and they almost become a conductor. Okay? And they generate sufficient. So, and if you manipulate the physical parameters, they generate sufficient amount of current which could power a low power electronic devices. And the proposed model is shown here, and these are some of the results, and this is the proposed model out here. And you could do the similar things with porous materials. So, I will close in here, and I will request you people, please go through these papers. They are very interesting papers from Jacob Ishay now, and there are a lot of room for development and understanding, for even a better understanding that how different kind of biomaterials could be utilized, could be an answer for our quest for clean and green energy. Thank you.